Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gil Eapen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gil@eapen.info. My guest today is Professor Robert Murphy, who is a professor of computational biology, biological sciences, biomedical engineering, and machine learning at Carnegie Mellon University. He is also Honorary Professor of Biology at the University of Freiburg, Germany, Fellow of the IEEE and the American Institute for Medical and Biological Engineering, and a senior member of the International Society for Computational Biology. He founded the Computational Biology Department at Carnegie Mellon and served as its head from 2009 to 2020. His research interests include machine learning of image-derived models of cell organization and analysis and modeling of protein location changes across cell types and diseases. Welcome, Bob.
Thanks, Gil.
So your passion at CMU is what you call automated science, which is the practice of scientific research without the need for significant human intervention. And the goal is to develop self-driving instruments, you say, along similar lines to self-driving cars. Could you explain a little bit about what you mean by automated science?
Sure. To do that, the starting point for me is going all the way back to when I first decided to become a scientist. I was 13 years old and I read a book by Isaac Asimov on the genetic code.
Yeah.
And that it was a fascinating book. And what appealed to me, what's sort of a premise of that book is that we were starting to learn the rules, the codes, the laws, such that we would have the kind of understanding of biological systems that we had of mathematical and physical systems.
Yeah.
And that really was extremely attractive to me, and that's what led me to go in the direction that I've gone. But while I was still in graduate school in the 70s, the whole paradigm of learning the laws, learning the rules started to really crumble. And what I remember really well from that period was the discovery of reverse transcriptase, which is an enzyme that turns RNA into DNA.
Yeah.
And the central dogma is DNA makes RNA makes protein. And this was turning that on its head. And that was followed by many, many, many other examples of biological laws that we thought we had learned, especially starting in the 50s and 60s, that just turned out to be basically suggestions. And that really started the whole community to think about what do we do, in terms of biological research or biomedical research, given that discovery. And I'm sure, many of your listeners are familiar with that. And in addition, what we learned is not only weren't there any rules, but biological systems are what are called complex systems, systems that are the behavior of which is not apparent from their individual properties, right?
Mm-hmm.
Systems that have emergent properties that you don't see of the parts. And so in that way, they're very different than things that people make, like cars. You can build a car and take the car apart and figure out how all the pieces work, and put it back together and have an understanding of how the car works. Well, biological organisms are not like that. And so we began to have this understanding, now many years ago, that biological entities were complex systems. And so there was a rise of what's called systems biology.
Yeah.
And systems biology tried to take a more holistic approach, and move away from the reductionist approach that had been used previously, the idea of taking things apart into individual pieces and trying to figure them out, to try to characterize complex systems. But what I believe was missed through most of that was that when you have a reductionist system, the number of experiments, we'll call them, that you need to do in order to understand a reductionist system is just the number of pieces. You do one experiment per piece, or ballpark, and now you've understood what each of the pieces do, and then you're done.
Right.
But when you have complex systems, you have interactions among them, and so you have to do many combinations. You have to study combinations of things, how they work, how they affect things. And so what began was a lot of experiments in which large-scale studies were done to see what are the effects if I, delete this protein or block this protein, or add this drug, and then try to use that to build models of those complex systems.
Yeah.
And so the idea was to replace laws, biological rules, which there didn't seem to be any of, with computational models.
Mm-hmm.
And the natural temptation that happened at that point is that investigators would do a huge amount of work to collect a lot of data on a very small subset of genes or drugs or cell types or whatever it might be, and then try to build a predictive model of what the properties would be of those things for the rest of the things they hadn't measured, the rest of the drugs, the rest of the proteins.
Right.
And there was then, to some extent, an understandable but a mistaken idea that arose, which was, well, now that we've built this model, we have to validate it. We have to show that it's correct.
Mm-hmm.
But that forgets the first rule of modeling, which is all models are wrong.
Right.
And there is no way to prove an empirical model.
Mm-hmm. Mm-hmm.
That's the very simple fact that if it is an empirical model, if it is not based on underlying rules, you never know whether or not any individual prediction from that model is correct. You might have a sense of most of them being correct, but what ended up happening is that reviewers for papers would ask, validate your model.
Right.
And investigators would say, all right, well, let's see what we can do. We'll take the highest confidence prediction from our model, and we'll do one more experiment on that.
Mm.
And if it's right, we'll consider our model to have been validated, or maybe we'll do 5. But all that does is prove that specific prediction was correct.
Right.
Does nothing to prove the model, because you can't prove an empirical model. Now, that's kind of brings us to the idea that what experiments are for is not to prove empirical models, but to improve those models.
Mm.
And therefore, we need to move towards a paradigm, which is we are continuously, iteratively building the best model that we can and using those models where we desire to, without having to try to pretend that we're proving the model. We just say, this is the best model we have today. Let's use it.
Right.
But we don't need to try to prove it. Now, how do we go about doing that?
So,
Well,
let me ask a quick question on that, Bob. So there are two issues there, right? One is biological systems. We haven't been really be able to create very specific rules as to how the systems work. They're very complex. They're very non-linear. Lot of combinatorial issues there. So the design space, if you will, is very, very broad and wide. And it seems like what we typically do is to take subsets of this design field and come up with a model, and we'll have some rationale that that model is working or not. And if it's not working, we'll go find another model and another subset. And it doesn't really solve the problem. Do I understand that correctly?
Yes. So there are two pieces there. One, is it just that we haven't learned the rules? And I don't think that's the case.
Yeah.
And can we try to build models by simplifying systems and then building models that capture the properties of the simple system and then expect that they're going to apply to a larger system? And the answer there I also think is no. So that trying to break systems down into simpler systems is often something that will not end up leading to a good predictive model.
Right.
Now, we can talk about why, it's reasonable, or why we can think that there aren't any biological rules. And that's by just thinking about the challenges that evolution faced over millions of years of how do they adapt an organism, or how can an organism adapt to a particular changed environment or new predator or whatever it might be. That organism had a set of parts already in it,
Mm-hmm.
and basically it just had to try to modify those parts to to accomplish better survival or whatever is needed. And this is a little bit like, if you've ever seen the movie Apollo 13, there's a scene where the NASA engineers, they're trying to build a CO2 scrubber for the capsule, and there's a scene where a bunch of NASA engineers, the guy comes in and dumps all the parts that they have in the capsule,
Mm-hmm.
and says, we have to build a scrubber out of this. Well, that's what evolution has been doing for millions of years, is taking the set of parts and finding new roles for them, modifying them slightly, connecting them together in ways they weren't connected before. And while you might have said prior to that, oh, this piece is for X, that's no longer the case. That piece is now doing X, Y, Z, and W.
Yeah, and the additional complication there also, Bob, is that when you make that favorable modification, you also pick a bunch of errors. That is completely uncorrelated, right?
Sure, you get, yes, and it makes it more and more complex, and therefore, what may have been rules in the very earliest days of life quickly became just suggestions. So, what do we do about that? Well, fortunately, there are a lot of automated instruments that have been built, beginning largely with the Genome Project, but during the last 20, 30, 40 years of systems biology, automated lab equipment that has been built both for clinical labs, but also for biomedical research labs, that enable experiments to be executed very efficiently, very reproducibly on large scales. And that has been essential to the rise of systems biology, to be able to make many, many measurements.
Yeah.
But what we are typically doing in those cases is picking a subset, a little, if you want to think of it as a piece of a large matrix, and doing all the experiments in that space,
Mm-hmm.
and then trying to make predictions for the rest of that space.
Right.
And that's where the modeling exercise comes in. And if you just take that small chunk, you're probably not going to get a very good predictive model for the rest. So there is a technology from machine learning that is appropriate for this type of problem. And that's what's called active machine learning.
Yeah.
So what we're talking about is a case where we have a space that is too large for us to do all the experiments. When you have complex systems, basically anything can interact with anything. So if you're trying to model the behavior of that thing, you might have to do, essentially untold numbers of experiments and all the combinations of 10,000 genes, or many, and then of course there are many other things, millions and millions of possible drugs and so on. Yeah. So in that case, we need a way that we can explore a subset of that space, and that's what active machine learning is about. And so automated science, well, it took me a long time to answer your question. Sure. Automated science is about combining laboratory automation, which is those instruments that could do or can do biomedical experimentation, usually under computer control and usually very reproducibly, and again, usually at high rates, to combine that laboratory automation, which up until now has been controlled by people. So scientists are saying, do this grid of experiments, please, instrument.
Right. Yeah.
And people are not good at deciding what experiments to do in an enormous experimental space in order to build the best possible model. That's just not something we are ever going to be good at. Right. And that's what active machine learning is about. You build a computational model, and then you use that model to decide what experiments should I do next in expectation that they will improve my model.
Right, right.
And a really simple way of thinking about active machine learning is what's called uncertainty sampling, which is to say, do the experiment that your model has the least confidence in predicting.
Yeah.
And that will improve upon your model, right? Now you'll at least know, what happens for that particular kind of thing. And hopefully you can reason from that, or you can predict other things from that. So that's what automated science is from our perspective.
Okay. Yeah,
combination of laboratory automation and artificial intelligence, active machine learning.
Right. Yeah, it reminds me something, Bob. So this is a different problem in the physical sciences area. So my graduate thesis at Northwestern in the mid-'80s was an expert system that attempted to build design intuition in engineering students by helping them interact with a computer. So you'll see, you'll get a chuckle out of this. So the problem we tackled was to teach a student how to create an engineering design with the lowest cost for a presented problem. So that design space is not huge, but there are features in it, and it spans quite a bit. So typically students learn this in the field, how to minimize cost by repeated experiments, right? They make designs, sometimes they make mistakes, and over time they get better at it. And and the idea here was, so this was obviously crude technologies in the 80s. You can imagine IBM PC and a language called Pascal, and I suppose it's dead now. But it's a rules-based system. So essentially, student sits in front of the computer. There's a design problem. Student makes some choices, and then the machine basically gives guidance as to which direction to head in that design space, right? So a fresh student or a naive student might end up with an exhaustive search of the design space before he or she finds the right answer. Here, the idea was we can substantially reduce that time to reach that minimum by giving the human, in this case, a student, feedback by the computer to do that. Obviously, it's a lot easier to do. No machine learning there. It was all heuristics and rules-based. A lot easier to do in the physical systems space, as you say. But when you get to biological systems, I guess the issue there is we don't have nice rules there. So what you're presented with is a very broad set of features and combinations of features. And oftentimes, not a lot of data, right? Not a lot of those experiments have been done yet. And so you can't really deploy conventional machine learning, which is typically deployed when you have a lot of data. What you have in biological sciences really is a problem definition and really a definition of the design space that you want to explore in.
Right. And that is really counter to what you hear a lot of times about biology, which is, oh, we have to have better computational algorithms to deal with the enormous flood of biological information. Well, in some terms, there is a large flood of information. There's a large amount of information. But if we look at that amount of information relative to the amount of information we could have relevant to that very large experimental space that we possibly could have, it's a tiny, tiny fraction.
Right.
Big data in some sense, in some absolute sense, but it is tiny data in a relative sense. And that's the reason why we can't just brute force it. Right? It's a little bit like playing Battleship.
Yeah.
You have, say, for example, think of a battleship's grid as trying to explore the effect of different drugs on different targets within the body, right? So you have a matrix of drugs versus targets, or your battleship grid. If you play Battleship by going A1, B1, C1, D1, you're never going to win.
Right.
But that's what we are doing now. That is what we do is we do A1, B1, C1, D1, and then try to predict the rest of the board.
Yeah.
Where the AI approach, the active machine learning approach, would be to sample that space and try to build a model that would allow you to do almost random access. It is at random access sampling, which is how an experienced Battleship player will play.
Right.
And so that's really where we have to move away from, that paradigm that we know, as you were saying before with respect to your example, moving away from that paradigm when we know the design principles that there should be, where instead we just let it be data driven and then we use the models to decide what data to get.
Yeah, and the difference in automated science, your approach, Bob, is there are things like active learning and reinforcement learning. In active learning, the idea is really kind of introduce humans back into the process to relabel some of the mistakes the machine has made and make the models better. Automated science is a totally different approach, right? You are basically removing the need for a human in that space.
Yes, but in active learning, the human is often used as the term is used there as an oracle, but there's nothing about active learning that requires that oracle be a human.
Oh yeah.
And what we've done is replaced that human with a very accurate machine, a very reproducible machine. Right. And but it is the exact same algorithm. And just instead of asking a person, what do you think would happen if I add this drug, to this cell line? Instead of asking a person to do that, we ask a machine. Well, tell us what actually did happen.
Right.
And it also represents a very nice mixture between purely experimental approaches and purely computational approaches. So there are people who have tried for many, many years to just simply build computational models without relying upon that iterative experimental input. And this automated science is the marriage of those two. It's the marriage of the laboratory automation, the large-scale experimentation, but doing that more intelligently.
Yeah, I mean, the beauty of that is, we believe humans are pretty good at looking at large amounts of data and making decisions. But what data is telling us, it's not actually true. Another example, my firm ran a hedge fund in the mid-2000s, and it was a machine learning and a bit of reinforcement learning based, fully automated system. But more importantly, what we learned was that humans are the worst in making financial decisions, including trading decisions. But if you talk to folks who do trading or financial decision making in Wall Street and elsewhere, they will tell you they are really good at what they do. But if you look at the cross-sectional data, you can see, machines are far superior to humans when there is lot of dynamic data. We never evolved to make stock trades sitting in front of a computer, and
Exactly.
we're kidding ourselves, to say that we are good at it.
And there's an analogous case from more than about 30 years ago. We started working on using computer vision, machine learning methods to automate the identification of where within a cell a particular protein was found, which you would normally do by using fluorescence microscopy. So you use fluorescence microscope to look where a particular labeled protein is within the cell, in order to be able to say, okay, that's in the nucleus, or that's in mitochondria, or that's in lysosomes. And that task was done by people. So that you would label that protein, take a fluorescence micrograph, show it to a person, and the person would say, oh, that's a nuclear protein, or that's a mitochondrial protein. Mm-hmm. And we, realizing that there are so many proteins that need to be analyzed in many, many different tissues, thought that this would be something we should automate. And so we used machine learning approaches to train a system to be able to distinguish using regular supervised learning between different subcellular patterns.
Yeah.
Well, when I first started talking about that in meetings, everyone would come up and say, that there's no way you should be trying to do this. Humans are so much better at this task than people are.
Right.
So we then did an experiment where we actually tested how well the human does at that task. And just like you were saying in the financial case, people are terrible at it.
Right.
And so it's just, we have to recognize the things we're good at and not try to do the things we're not good at.
It's sort of a paradigm change, right? So I always believe that there is a lot of automation out there. There are a lot of, kind of machine learning, deep learning applications. And if you introduce humans into a process, let's say it's 95% automated, and then you introduce a human to take care of the 5%, you have human as the weak link in that whole process, and it's going to break. You look at airplanes. These airplanes are very highly automated machines, and almost all the accidents that we see in the air are related to human error. So in the cockpit, really the weakest link in the cockpit is the human, right? And so we might be getting to a point that this idea that humans are very good at certain things, and hence when the machine fails, we need humans to take care of it. I think it's a false notion.
Right. Yep. And instead, you just help the machine get better by having it do more experiments and have more to learn from. Right.
Do more experiments. And in your case, and again, going back to the automated scientific experiments, we cannot really do, kind of an exhaustive search of the design space. So you need an intelligent machine that's able to do that in a very efficient fashion. I think that is the trick, right?
Yes, absolutely. Absolutely. And now the natural question though becomes then, well, what is the role of human scientists in all of this in an automated science context? And there is an enormous role still to be played, but it is not doing pipetting repetitively, which is what a lot of biomedical graduate students do, for many, many years, is just manually executing experiments. We don't need them for that. And we obviously don't need them for picking which experiments to do in order to improve a model. What we do need them to do is to invent new technologies for measuring cells, or measuring tissues, or measuring any biological property, because Right now, we don't have any machine learning algorithms that can invent a new biomedical research technique,
Mhm.
right? And so we need people to do that.
Yeah,
But
so it is. Sorry, Bob. So
oh, go ahead.
would you say, so, I think about, so would you say the activities that should be delegated to a human has to have a very high creative component to it? So
Absolutely.
anything that is repeatable, anything that is automatable, we can I think conclusively show machines are going to be a lot better, right? So that should give us activities that humans should really be focusing on.
Absolutely. And anything that is repetitive or anything that is high dimensional,
High dimension,
because
yeah.
humans can't hold high dimensional spaces in their heads, right?
Right,
But
right.
but if you're inventing a technique, that's a very creative and focused activity, and that's certainly something that people are the only ones that can do that now. Now, there's another thing that we need people to do, and that is if we go back to the analogy of an automated science as being equivalent to a self-driving car, right? We call it self-driving instruments.
Yeah,
The instruments not only execute the experiments, but they decide how to execute them and how to build a model. Um, when you get into a self-driving car, it doesn't decide where you want to go.
right.
You tell it where you want to go. So we still need people to prioritize what kinds of models, what kinds of systems are the highest priorities for us to study, whether it's in order to address particular diseases or to prevent particular syndromes, whatever it might be. We still need people to make those decisions about what's the goal, where am I going? And
Yeah,
so those are the two things that scientists and society needs to be able to do, but not the rest of the stuff that can be done in automated science.
yeah, I want to push on that a little bit, Bob. So I want to get your perspective on this. So is it possible that, if you go down this track in terms of automated science, is it possible that the machines will provide insights back to the human that we haven't really thought about? So, in your analogy, in terms of self-driving cars, we don't tell the car where to go, but presumably, when the autonomous vehicles get to level 5, one could envision a situation that the vehicle determines where to go for you because it can learn what you want to do. It can anticipate what you want to do. There are two different things there. One is sort of learning, the other is the machine making errors, perhaps, but then those errors result in some sort of insight for the human. That's also possible, right?
Well, I definitely agree that for example, the self-driving car could learn that if you get in the car with a bathing suit on, that you want to go to the beach,
Right. Yeah.
and it could learn to help by your saying, um, you know, take me to the nearest movie that's starting within the next 20 minutes.
Yeah.
Right. So there are plenty of ways in which it could help you decide where you want to go. But when you're talking about the goals of automated science, what you're talking about there is, which types of diseases should I try to find cures for or find therapies for?
Yeah.
Right. So the kinds of things, because we can't do all of them at the same time. So that's,
Right. Right. Right.
and those are very human priorities, right? To say, well, we're going to prioritize this. For example, right now, we would say we're going to put a lot of priority on COVID-19 research, right?
Yeah.
And that's something that we, as scientists and as a society, make that decision.
Yeah, it's this is sort of may not be practical, but very conceptually though. Even when we look at those decisions, how to prioritize resources, where to focus on, humans haven't made very good decisions. For example, antibacterial is a good example of this. COVID-19 is a good example of this. We tend to sort of fall into creatures of habit. I'll give you another example. I was in a large pharmaceutical company in the mid 90s, and I was on the kind of the business side. So we created an expert system that looks at the entire portfolio of products the company was developing, over $4 billion per year invested into products. And the expert system, we had to actually predict what's the likelihood that a product is going to succeed, given the characteristics of the product. So things like class of chemical, disease area, geographical location, number of projects adjacent to that project, project leader, and so on. And what we found was that we can actually reasonably predict what's going to happen to that compound, right? So what that tells me is that, there is a process that is sort of stale and stagnant in organizations, even in R&D organizations that are supposed to be, driven by creative processes. It becomes more like manufacturing processes that are highly repeated with a set of heuristics that are very limited. In which case, I think disrupting that using machines could be quite beneficial.
Yes. So I'm not suggesting, which is the current, like your experience relates to the biases that pharmaceutical companies may have of saying, well, our last 3 good drugs were made on this backbone, so let's use that.
Yeah.
Right? Or I have a hypothesis that this kind of thing would work well for this. I'm not talking about that kind of guidance, because that is you're absolutely right. That's usually wrong.
Yeah.
And one of the reasons we have now, we're approaching negative return on investment for drug development.
Right.
And so, because most of the time people are guiding the process and they're mostly wrong.
Right.
And so, but the question of whether we should put our efforts into a drug for cancer or for diabetes or for COVID, those questions I don't think a computer can answer.
Right. Right.
That's the part
Yeah. There's a horizon problem there. Yeah. I mean, this is a different problem altogether, which is what are the incentives in the system and how does society make those decisions? And that's a whole different problem that machines won't be able to solve. But I always wondered, even those processes that are kind of getting stale, introducing machines could disrupt that. I don't think machines are going to take over decision making, but at the very least, it can disrupt, maybe give some insights back to the human, saying, I want to just maximize profits in the next 2 years, and that's my resource allocation paradigm. And maybe machines can show that that's not a good thing.
It's a very interesting idea, and certainly very timely, given as the trend has been that fewer and fewer drugs are being developed successfully, and they're costing more and more. I think there's a disruption. Is needed for sure.
Yeah, and COVID-19, for example, I don't know if you're doing any work in that area. I know that there are a lot of computational approaches they are trying to solve this problem. But this may not be a single problem, right? We may have a series of problems coming in the future. So then there is a question around, there has to be some process, some kind of a new process for us to continue inventing new things. And I wondered, are you doing any work in that area?
Well, I'm not doing anything on COVID right now. But I do think that you're right that it may end up being way more complicated than we thought. And one of the things that automated science can do is to allow us to build larger and larger scale models that will take into account more and more things. And then it may be a matter of luck as to whether your current large scale models happen to have any insight in a particular new disease. Right. And one of the problems with COVID is that it really seems to have hit a niche that we didn't know very much about. But that doesn't mean that you might not be so unlucky the next time that, the next thing that comes along, there is relevant information in a structured way rather than in the form of what people think are good solutions, but rather data-driven and predictive model-driven.
Yeah, so there are some attributes here, right, Bob, in automated science that's a bit like DeepMind, Google's DeepMind. So if we have, let's say, we have wasted computing resources that are sitting around, not doing anything, are there opportunities in this approach to say, let it basically go explore spaces, even without any, kind of specific objective function?
Yes, so that gets into the whole guts behind active learning and AI in terms of what are the strategies to do active learning.
Yeah.
And exploration is clearly an extremely important aspect of that. But what you're doing typically in active learning is trading exploration and cost, right? And so when you can do a quick calculation, with idle cycles, that's one thing. But if you actually have to do a physical experiment, that's something else, right? Now, there's certainly need for computing in order to try to build and improve upon the models to choose that next experiment. But it's not like we have excess laboratory automation capacity sitting around that idle, right? And my suspicion is, well, actually, I shouldn't say that, because there are places that have automated laboratory equipment that do sit idle in between large cycles of doing, they do a massive set of experiments and then they spend 6 months analyzing the data and the instruments are sitting idle. So there may be something like that.
Yeah, especially in life sciences context. Obviously pharmaceuticals and biotechnology companies have made a lot of investments into these types of instruments. And like you say, there is sort of a sequential process that they go through: experiments, data analysis, decision, experiments, data analysis, decision. And those cycles, like you say, may give a lot of idle time for the instruments. Then the question is, are there ways to? I know that the search for extraterrestrial intelligence. They try to use kind of excess computing power, right? They took the data and gave it out to a lot of computers.
Right.
I wonder if there's something like that that could be done. That, I think it's much more complex, right? These instruments have to be set up. It's not like a computer. Yeah.
But laboratory automation has gotten to the point where you can run, and pharmaceutical companies do this, where you can run a lab 24 hours a day generating experiments. And with an automated science approach, we can shorten that cycle time you were talking about. So instead of collected a bunch of data and then, wait 3 months while you analyze it or 6 months while you analyze it, that process can be happening continuously so that you're not doing monolithic experiments and then monolithic analysis, but rather doing that iteratively, continuously.
Yeah, another thing I wondered about is, again, in a pharmaceutical context, maybe this is something they should do in parallel. So there is a status quo, status quo process that's largely driven by, not largely, but driven by humans, let's say. Perhaps this type of a process is in parallel to that. Recently talked with another guest about, we are seeing two types of intelligence, right? And I would love to get your insight into this. So the way that machines are building intelligence appear to be very different from human intelligence, very different, right? They need large amounts of data. They do some classifications. They do some unsupervised machine learning type thing. And so we might be coming to a point that we have machines become a sort of a different species, right? So its intelligence is quite different from human intelligence, both in construct and format and process, which would then imply that getting that diversity into R&D processes could be quite beneficial.
Yes, although you're starting to get outside the scope of my my thoughts.
Sounds.
I do want to mention, though, in terms of parallel.
Yeah.
One of the things that that I believe is needed is that more people need to be hired into pharmaceutical and biotech industries who are aware of this paradigm of automated science and can execute it.
Yes.
And that's one of the reasons we started a master's program in automated science last year. And to be able to train people in this intersection between laboratory automation and machine learning.
It's a very complex.
So go out and go ahead.
No, I was just going to say it is a very complex set of skills because you need to understand the domain really well. And then you need to understand, the computing aspects of it and the instrumentation aspect of it. So it is really a very, very complex skill. And I agree with you, sorely lacking, I think, in life sciences companies.
Yep. And we already have the first year of students have completed their first year, and they'll be graduating in May. So We're looking forward to where they go.
Excellent. So in conclusion, Bob, when you look forward next 5 years, in your domain, automated science and more generally, artificial intelligence, what are your expectations? Where do you think we will have the biggest, not discovery, but innovation, the biggest innovation that is practical?
I think that it will come from approaches that are not based on hypotheses or human intuition or somebody has a gut feeling this is the way to go. More and more, they will be driven by computational models that discover things that nobody had any prior inkling of. And that's the transition that will happen, is to people being comfortable with that. The heads of pharma and heads of biotech will be comfortable with the idea. No, I don't need to have the world's expert on XYZ to tell me what I should do. I can determine that empirically and use predictive models for that. I think that's the biggest thing that I expect to see over the next 5 years.
Yeah, it's a really big change. It's a cultural sort of transformation. And
absolutely.
It looks like technology will be far ahead of sort of the organizational changes that we need to make that happen. But I fully agree with you. I think that is inevitable, just a matter of time.
Yep.
So this has been great, Bob. I really appreciate the time that you spent with me, and good luck with continued success in this area of automated science.
Well, thank you very much. It's been a pleasure talking with you. Thanks for the opportunity to talk to your audience.
Thank you. Bye.
Take care. Bye-bye.